There was one thing that I forgot to mention to you about the epitopes and so forth. I just remembered coming in here today. There's something called hapten. It's kind of difficult to understand, but if you listen, you'll be fine. We talked about epitopes are those regions on an antigen, like a foreign protein, that elicit an immune response. Not all epitopes do, but the ones that are obviously immunogenic. So we said, for example, we have 3 epitopes on this protein, for example. They're basically amino acid, a stretch of amino acid sequences. So this is protein. Epitope number 1 may not elicit an immune response. And let's say epitope 2 does. So your immune system will produce an antibody against, for example, epitope 2. And we talked about the bigger the protein is, the more immunogenic the protein is. Why? Because it'll have more epitopes. You can overcome this problem by simply providing more epitopes. So if you have epitope 4, 5, 6, 7, 8, 9, 10, you'll have more chance of an immune response. If you're only dealing with 1 or 2 epitopes, there's very likely, the likely chance of producing an immune response is very low. That's why molecular weight is a huge factor in immunogenicity. The bigger something is, the more likely you'll have epitopes that actually respond, that your immune system responds to. Here's the interesting thing. Watch this now. I know epitope 2 your immune system responds to epitope 2. But look at what happens. Watch. If I take epitope 2 out of this, meaning I just used epitope 2, that's it. If I inject that in you, you will not get an immune response. That is now called a hapten. Because it's too small. That's why. So it looks like epitope 2 is immunogenic only when it's part of the whole piece. You see that? So this molecular weight thing really plays a huge role to stimulate your immune system. So epitope 2 on its own, even though it elicits an immune response, but on its own, it will not. And that's called a hapten, H-A-P-T-E-N. Now, the interesting thing is this. Sometimes we get an epitope like this. We know it's immunogenic when it's part of the big protein. But for some reason, we can't isolate that protein. For some reason. It's too difficult to isolate. It's not stable, I don't know. For some reason, we can't get it. So we cannot use it as a vaccine. So now what do? Okay, we just cut that piece out. Well, that alone does not produce an immune response. So now you're caught in a problem here. You cannot purify the full-size protein. And if you do genetically engineer where you're only making epitope 2, that's not going to stimulate the immune system on its own either. So now what do? They figured out a way to do this. They attached this one to something called a carrier. And that carrier molecule, look what it does. It tricks your immune system. It says, I'm actually this one, even though it's not. Your immune system says, well, that carrier added on the molecular weight. So a lot of times when we have a hapten, on its own does not produce an immune response. We attach it to a carrier molecule to simulate the molecular weight, to satisfy the need for high molecular weight. Your immune system will now respond to this one. But on its own, it does not stimulate the immune system. So you have to attach it to a carrier molecule. So you may see that as part of this discussion in the soft chalk. Yes, sir?
So how does it treat the carrier?
Huh?
How does it treat?
No, this is a neutral molecule. It doesn't do anything. It's going to respond to this guy. All right, so I thought I would add that in there because I think, if I remember right, there is a discussion on hapten in your soft chalk. Okay. This is where we left off, right here. Let's talk about antibodies real quick. We'll talk about the different classes of antibodies. I will reinforce what happens with the T cells and the B cells one more time. And there are a couple of slides at the end that we'll go over, and Unit 10 will be done. Your antibodies, they're actually made up of 4 polypeptides. 2 long ones and 2 short ones. So the short ones are called light chains, and the long ones are referred to as heavy chains. They are attached to each other in this Y-shaped structure using disulfide bridges. So these are cysteine, the amino acid cysteine. When cysteine faces another cysteine, this is not what this C is. There's an amino acid called cysteine here facing another amino acid called cysteine. That creates a disulfide bridge under the right conditions. So there's another one here, 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 here. And that's how you attach all 4 of them together. This C stands for constant region. So the sequence of the amino acids here and here and here and here, those are referred to as the constant region. They don't change. Among your antibodies, they don't change. But the change is here in the variable region. So remember when I talked about, when was it? Thursday? Thursday, right? Yeah, Thursday. I said that you get a shuffle, and you're born with all the different shuffles. Hopefully you remember me saying that. This is a product of that shuffle, the variable region. So one of the shuffles would produce a variable region that recognizes, for example, the blue cap pathogen, like I used in class last time. But you gotta understand something. Whatever this shuffle is, this shuffle will be the same. So if this recognizes the blue cap pathogen, this one will recognize the blue cap pathogen, the same one. You will never have a variable region or a shuffle here that is different than here. That's a different antibody. So these antibodies, that's how they gain specificity. Why is this antibody specific to the blue cap pathogen? Because it has the shuffle, by random chance, that recognizes that blue cap. You want to recognize a red cap pathogen? Yeah, then you have to have a different shuffle. So that's called the variable region. It is also referred to as the antigen binding site, because that's where the antigen will bind. It'll be the same antigen binding here as the same antigen binding here. And there are different kinds of antibodies. As you see here, there are monomers, there are dimers, there are pentamers. So there are different kinds. There are short ones and longer ones. That's a lot of information, I know. But what I'm about to say is what you need to know. The IgAs, those are the dimers. they are linked to each other through this thing called the J chain. That's like the glue. And that's how they stick together. The most important thing about the IgA antibodies is that these are antibodies found in secretions. So you're going to find these IgA, for example, in breast milk. And this is the main antibody that a mom will provide for her infant during breastfeeding. It's also found in other secretions, but one of the most important ones would be, of course, breast milk. So IgA is the antibody. It is a dimer linked by a J chain, found mainly in secretions. All right, IgM. IgM is a pentamer, also bound through J chains. Same thing here, see, J chain. This antibodies, the important thing about this antibody is this. When you first get exposed to a pathogen, let's say the first time you got sick with COVID-19, it's IgM that initially came on the scene to help you. Upon first exposure to any pathogen, IgM is what initially protects you. A little later, IgG is produced. And you'll see that there's a diagram for that at the end of the unit 10. So you gotta remember, first exposure is clearly recognized by an IgM over secretion compared to IgG. So your IgM curve will be high compared to the IgG. You'll still get both, but you'll get more IgM than IgG. You see a graph like that on, for example, an exam on Thursday? Oh, that's primary exposure. That's the first time you came down with this infection. You're going to initially be protected by IgM mainly, then IgG. Then you can tell what happens now, hopefully. The second time you see the same pathogen, it'll be mainly IgG protecting you. So IgG is for long-term protection. IgG is made later in a primary infection, but it is the main protector long-term after that. So secondary exposure to the same pathogen, if you look at the curve, IgG will skyrocket. You'll still get some IgM, but your IgG will take off. You know, you're gonna be dealing with this as a medical practitioner. A doctor sometimes orders a test and asks the lab to test for, do you have IgG protection against this pathogen, or is it mainly IgM? That's another way of saying, was this your primary exposure, or was this a secondary or a tertiary or more exposure? So they can test to see, are you mainly fighting this current infection with IgM or IgG? If it's IgM, that tells them you just got it. This is the first time you saw it. They're most likely going to be expecting IgG, because most likely it's probably the second time, third time, fourth time seeing this pathogen. So if they see IgG, that means whatever that pathogen is, you've had it for a while, or you've had it multiple times. Okay, this guy here, IgD. That's the antibody. Watch this now. That's the antibody that is on the B cell. That's this right here. The IgD is the B cell receptor. and I mentioned it on Thursday, and I will go over it again. When the B cell differentiates into the plasma cell, this guy, you see, it will not have the antibody on the surface anymore. It's a completely different cell. It came from the B cell, but it's a differentiated B cell. So it's different. The main difference that you need to worry about is that the plasma B cell will secrete the antibody into your blood, compared to the B cell, it has the antibody on its surface. But now you know that that B cell receptor is an IgD. If you are suffering from allergies, and some of us are, you're gonna now blame it on your IgE. So, like everything, like all the other antibodies, watch this. You produce an IgE antibody that has a variable region that happens to recognize, unlucky for you, pollen. And that's why you would have something like seasonal allergies. Every time the flowers bloom, your life becomes miserable. For a little while at least. So, again, you just happen to have a shuffle that happens to recognize things like pollen, peanut butter, right? All the common allergies that we suffer from. So it's on your IgE, and once it attaches to that, what is referred to now as the allergen, then it activates the allergy response, and the nasal discharge, and the tearing, and the stuffiness, and so forth, because you release histamine. So IgE mediates allergic responses. It's also involved in fighting parasitic infections as well. So I did not talk about half-life because that's just too much detail. So we give you the information, but what I just mentioned is the most critical information that you need to know. And you'll have it on video to go over it again in case you weren't able to write everything down. All right, we did this already. These are the B cells and the T cell receptors, the MHC1, MHC2. All right, here we go. In case you missed it on Thursday or you heard it, but you did not catch it the first time, I'm gonna do it again. The CD4 surface receptor here, that's called cluster of differentiation 4, variety 4. And I told you there are hundreds of them. And I decided to limit it to only 2. All you need to know is 2 of them. CD4 versus CD8. CD4 is the signature of a T helper cell. On the surface of the T helper cell, it has CD4. It has other CDs, but it's unique in the sense that it is the only T cell that has CD4. So I'm gonna recognize this as a helper T cell by looking at the CD4. The cytotoxic T cell will have a CD8 on the cytotoxic T cell. So again, the cytotoxic T cell has other CDs, but it's the only T cell that has CD8. So I'm gonna use that to identify. All right, so what happens with the helper T cell? Now again, this is a helper T cell because I see this CD4. What's gonna happen is this: the antigen presenting cell, which is the macrophage, dendritic cell, and possibly a B cell will process the antigen. So it'll phagocytose the antigen, the pathogen. Obviously, it's going to kill it with the lysosome, but then it will process a piece of it and send that antigen to the surface on the MHC2. So any antigen other than viral will be presented on MHC2. Viral antigens are presented on MHC1. There you go. But that's a different story. So we're going to exclude viral antigens for now. So all other antigens other than viral will be presented on MHC2. Presented to who? To the boss of the immune system, the helper T cell. How do I know it's helper T cell? It has CD4. Now the idea is this. Why this helper T cell? Well, this helper T cell has a T cell receptor with a shuffle. They also have the shuffles too. That happens to recognize this antigen. Remember, for every B cell that has a shuffle, there's a T cell that has the same shuffle. So there's a B cell that recognizes the blue cap pathogen, and there's a T cell that recognizes also the blue cap pathogen. So this helper T cell has a T cell receptor with a shuffle that recognizes this antigen. And then they come together. And this is called antigen presentation. Why is this step important? Because of what we classify the third line of defense as being acquired immunity, adaptive acquired. What does that mean, adaptive acquired? It means you have to activate the T cell. The T cell is there, but you have to tell it, hey, this is the bad guy, by presenting it with the antigen. And that's why we call this third line of defense adaptive acquired immunity. So once this happens, it activates this T cell. And why this T cell? Because it has that receptor that binds that antigen. We call that, if you remember, clonal.
Expansion.
Nope. Why did I select this one? So it's clonal selection. Among all the army of helper T cells, why this one? Because it has the T cell receptor that recognizes this antigen. So that's clonal selection. What you're going to see next is this guy is going to start multiplying. Part of the response is that it starts multiplying. Now with that one we called clonal expansion. Very good. And then we'll see all the different kinds of cells that it will produce and what they will do. Okay. The same thing can happen with the cytotoxic T cell. Now there's nothing on here that says that this is a cytotoxic T cell. How did I know that? It has CD8. Very good. And as soon as I see that, I know the antigen that will be presented is viral. The CD8 cytotoxic T cell will deal with the viral antigens. The other thing that tells me that this is a viral antigen is who's presenting it. MHC1. Yeah. So this cell is presenting a viral antigen on its MHC1. Why? Because it's infected. So if you look like a minute right before that, what happened is this cell got infected. It will process the antigen, the viral antigen in this case, knowing that it itself is doomed. I'm going to ask you a question. While it's doing this, and before it presents the antigen, it's releasing something. to help protect the other cells. What would that be? It's a second line of defense mechanism. Huh? Yeah, it's the alert. Alert, alert, there's a bad guy in the neighborhood. Huh?
This is how the other cells that like are infected. So you
Yes, yes, what's the name of that?
Tagging,
Huh?
tag something.
Huh? Say it.
Is it cytokine?
You can actually get it as a medication now.
Is it histamine?
It's okay. You'll go home and study, right?
Yeah.
Interferons.
Oh.
Interferons. So while this guy is processing the antigen, it realizes it's doomed. When I present the antigen to CD8, CD8 is gonna kill me. And I'll show you how it does it.
So it's
But before that happens, it will release, now we say it now? Interferons. That's the alarm. There's a bad guy in the neighborhood. Get prepared. He's gonna knock on the door. He might even enter the cell, but surprise him that you knew he was coming. And every time you get, whatever, the flu or whatever, that's what happens. You get rid of the cells that have been infected, and at the same time, the cells that were infected are protecting the healthy ones. And eventually, over time, you clear out the virus. You're sick for a little while, but you eventually clear out the virus. I think I heard a question somewhere. Did anybody? Yeah.
Is it only with the cells that present the virus that are releasing interferon?
Yeah.
Okay.
Yes. The ones that are already infected are the ones that are sending the email to all the neighborhoods.
And it's only
All
for,
the, huh?
it's only for viruses.
Yeah, virus. Interferons, viruses. Yeah. Now watch. Once this cell recognizes the fact that this cell is infected with the virus. Now I say, can you kill yourself and get it over with? Well, it's not gonna actually do that, because I make it sound as if it's easier than it actually is. I made it sound like, hey, kill yourself, and I leave, and no, it actually makes sure it's dead. It's not gonna let go until it makes sure it's dead. Look what it will do. Can't remember if I have a slide on this or not. If I do, doesn't matter. I'll just say it one more time. This cell will secrete a protein called perforin. Perforin. I mean, if you look at the word perforin, perforin comes from perforate, means to put a hole in it, to rupture it, to break it. And that's exactly what it's going to do. It's going to release a protein that puts holes in your cell membrane. And you'll be like, oh, it's going to lyse. No, that's not what it's doing. So what is it doing? It's opening the door. Because what it really wants to do is equivalent to this. Somebody opening the door and throw a grenade in this room. Blow it up. That's amazing. Grenade, the name of the other enzyme is granzyme. See, I'm trying to show you or tell you a way to remember it. But this is the analogy. The analogy is equivalent to this. Open the door, throw a grenade in there, and it will blow it up. Open the door, perforin. The grenade is the granzymes. Molecularly speaking, what actually is happening is this. The perforin opens the door, right? That's straightforward. The granzyme, when this cell puts granzymes in this cell, it activates apoptosis in this cell. granzymes activate programmed cell death. All your cells have this default program. When I'm bad, yeah, kill, terminate. It's amazing. It's actually amazing that all your cells have that. Hopefully they don't all use that program all at one time, because that would be bad news for you. But in a case like this where it's infected, yeah, you want it to activate that program. The granzymes flips the switch. It's amazing. That will not survive. Okay, questions on this? This might come back up again in another slide. I can't remember, but I think there is one. Now you got this one. We talked about this before. When we went through the list of the different kinds of antigens, the last one on the list was something called super antigen. Why is it called super antigen? Because of what it does. Watch. If you notice in the previous two pictures, the antigen was presented to the T cell, whether it's a helper T cell or a cytotoxic T cell. When you present the antigen to the T cell, besides the fact that you activate the T cell, you also ensure a tailored response. You say, I know you're about to do your job. I know you're about to attack, but do it nicely. Be nice. Be nice to who? To the host, not the pathogen. You don't want to be nice to the pathogen. Antigen presentation like this will produce a response that is tailored, meaning enough to kill the pathogen, but the host should be just fine. But then there are some antigens that bypass this. Look at it. The antigen itself, without the antigen presenting cell, goes and binds to the receptor. So you don't get this tailored response. What you get is a massive response. And that type of response will hurt the host. It can actually even lead to death of the host. That's how bad it can be. You do not want the antigen on its own to activate the T cells. Now there are some, like some exotoxins that do this. And those would be collectively referred to as superantigens. Superantigen is any exotoxin, for example, that directly binds to the T cell receptor and directly activates it without antigen presentation. The consequence of that is a massive immune response that the host may not actually survive. Yeah, go ahead.
I was gonna ask if there are any examples of but you just answered you said toxins.
Yeah, and exotoxins. There are different kinds of them that do this. Okay. The B cell. The B cell can also be, remember the B cell has a counterpart shuffle that can bind to the antigen as well. So antigen can directly bind to the B cell as well. The B cell can also phagocytose the antigen or the pathogen and present that antigen as an APC. Remember, B cells is also an antigen presenting cell. So the B cell can also phagocytose a pathogen, process it, and bring it to the surface of the cell. It has MHC2, and it will also have MHC1, but it will present MHC2 antigens to helper T cells but it will present MHC1 antigens to the cytotoxic T cells. Now, once the helper T cell is activated, how does it get activated? Through antigen presentation. It will release a whole bunch of things. One of those things is called interleukin-2. Interleukin-2 activates other helper T cells. By the way, the helper T cells, there's different subtypes. And I'm not gonna go into that detail. But it will activate helper T cells, it will activate the cytotoxic T cells, it will also activate B cells. It will activate the B cell that has the antibody that is similar to the shuffle of this one. So there's also clonal selection on this side too. And they all come in play, they all attack and proliferate to kill the pathogen. Among the pool of T cells that are produced, you will also get something called regulatory T cells. And we talked about it on Thursday. And if you listen to the name, regulatory T cell, that is a T cell that regulates the immune response. It keeps checking. Are we done? No. Okay, keep going. Are we done? Yes. Okay, stop. By the way, you will also get a regulatory B cell. So you'll have a regulatory T cell and a regulatory B cell that regulate the immune response. Among them as well, very important one, you will get memory T cells. That's important. Because you want to remember this event. If you survive it, you want to remember it. In case you come in contact with it a second time or a third time. And I mentioned to you before, that's what we take advantage of when we vaccinate. We'll show you that. So you'll also produce memory T cells and memory B cells. And the whole idea is to, of course, attack the pathogen and kill it. When you activate the B cell, again, it too will undergo expansion, multiply, proliferate, expansion, same thing. It will produce memory cells, regulatory B cells. But this is one of the other important things about B cells. A subset of the B cells will differentiate into a plasma cell. It will change and become a plasma B cell. Some places call it plasma cell, other places call it plasma B cell. It's the same. Then I mentioned to you already what the main difference is. The B cell has the IG, which one do you remember? Which one is this? IgA, M, E, D? IgD attached to it. But once it becomes a plasma B cell, it's now secreting that antibody into your blood. So what type of immunity is that? When you secrete the antibody into your blood. Humoral immunity, very good. You gotta understand something about antibodies, and it's gonna come in play soon. Antibodies technically do not kill anything, remember that. They tag things for destruction. Think of an antibody, like I told you before, as a Post-it note. One of those yellow Post-it notes. If I put a Post-it note on this chair and the note says, please remove, the Post-it note itself will not remove the chair. I'm just tagging it, waiting for someone to come in the room after I call them. There's a chair for you to remove. They gotta find out which chair it is, and they'll see the Post-it note. Similar analogy. And we'll show you what they do. So here are all the different kinds of T cells that are produced once the T cell is activated. Some of the more important ones are the memory T cells that I talked to you about, the regulatory T cells, and the fact that it stimulates also a counterpart B cell. That too produces a memory B cell and a regulatory B cell and the important plasma B cell. Does anyone want me to repeat what happens when after you present the antigen to the helper T cell and so forth, the sequence of events? Any questions? Because now we're switching topics. Oh, okay. So when we answer the questions, so hopefully you'll be able to answer those. If not, you ask me and I cover it again. All right, here's what antibodies do. Who produces the antibodies? Which one? The B cell. The B cell directly or the differentiated B cell? The plasma cell. Very good. You gotta speak up so I can hear you. So the plasma cell, it's not necessarily the B cell. It's the differentiated B cell. See how, you gotta be careful. Because on an exam like Thursday, I might have B cell on there. That's one of the choices. But the one that releases the antibody into your blood is the differentiated B cell called plasma B cell. Be careful. Or you just call it plasma cell. Now the idea is, what does this antibody do? It tags things for destruction. The closest an antibody will come to killing, listen to it, is this one right here. Is what's happening here. An antibody tags the bacteria for destruction, yeah? And obviously it's not just one antibody, it's a whole bunch of them. And they start interfering with the normal everyday function of the bacteria. The bacteria wants to go from here to here, but it can't. Why? Because it's got all these monkeys on its back, right? Called the antibodies. It becomes a little harder for it to move, for example. It wants to multiply, it can't. So this is, at the very least, the closest it will come to killing the bacteria. It interferes with the bacterial normal functions. So its binary fission might be slower than normal, its metabolic activity might be slower than normal, and so forth. So it's more of an indirect effect of potentially killing. But you want to kill the bacteria? Yeah. Watch this. This guy is a phagocyte. If I can turn him into a super phagocyte, then I would really kill the bacteria. Do you agree? I'm going to turn this guy into a killing machine. How? By tagging the bacteria like this. This macrophage can grab onto the bacteria much better. Now look at the two scenarios. One scenario is the bacteria does not have an antibody on top of it. So it might be a little difficult to grab onto, you see? But eventually it will grab onto it and phagocytose it. So without the antibody, it can still phagocytose it. But with the antibody, it will phagocytose it much better. We call that enhanced phagocytosis. Your textbook or soft chalk calls it opsonization. Opsonization, enhanced phagocytosis. So technically, you didn't do anything to the macrophage. It's still a macrophage. What you did is you put these grip molecules, the antibodies, on the bacteria that allows the phagocyte to grab onto them much easier and phagocytose them at a much faster rate. So opsonization is achieved through the antibody tagging of the bacteria, allows the macrophage to become a super macrophage. Now, there's no such thing as a super macrophage. It's called opsonization, enhanced phagocytosis. But who's actually doing the killing? It's the macrophage. Okay, we come over here. One other thing the antibody will do is something that you will hear about in, for example, at the time of war or war planning and all that stuff. Contain the enemy. Anyone who's planning to go to war or whatever, is the number one thing is to contain the enemy. Of course, then they can surround them and all that stuff. Contain the enemy. That's this one. Contain the enemy by making them stick to each other. Just think about it. Right now, you can go anywhere in the room. You can even leave the room if you want. Why? Because you're not stuck to anyone. If I want to keep you here, I can attach you to the two neighbors next to you, and then that person can be attached to the two neighbors next to them, and so we're all attached to each other. Okay, good luck leaving the room. We're not gonna go anywhere because we're stuck together. That's called agglutination. Agglutination. Contain the enemy. I mean, this is war, and it is so similar to actual war. If you contain the enemy, yeah, then you can focus your attention on them. The worst thing that you can do is ignore the enemy and have them go all over the place, and then wake up to fighting them. Are you kidding? Then you have a fight here and a fight here and a fight here, and you most likely you will lose. This is smart. Contain the enemy in one location and come at them with everything that you got, including opsonization. So this is technically not killing. It's just containing them in one area. Then this guy comes around and kills them. Or better yet, not better yet, or at least another option is this guy. The complement system. Contain the enemy. It also identifies the enemy. How do you identify it? With the antibody. And that's what the complement system wants. If you remember the complement system from the previous unit. The complement system, the C1, the C2, the C3, and eventually gets to C9. C9 puts a hole in all of these guys. So they're contained, so they're not going anywhere. They are tagged, so the complement system can also identify them. They have no chance against the complement system. On top of that, you're coming at them with opsonization. So who's actually doing the killing is the complement system and the macrophage. All right. What if you have a virus? The virus, you want to attack the spike proteins. That's why when you stood in line and got a vaccine for COVID-19, that vaccine was against the spike protein. Because what do you want to do? You don't want to fight the virus after it enters your cell. It's too late. You want to prevent it from knocking on the door to begin with. And that means attack their spike proteins. So your antibodies, the antibodies will be specific to the spike proteins on the surface of the virus. And that's a good thing. First of all, that's what it sees. Your immune system will see what's on the surface and attack them. It can't see inside the virus. So it will see what's on the surface and attack them, which happens to be a good thing because it's attacking the spike proteins. neutralizing the enemy. And that's why it's called neutralization. The antibodies that you produce that target the viral spike protein neutralizes the virus. But you gotta pick this up. Watch this. There are two kinds of neutralization. There is neutralization of the virus through the antibody binding to the spike protein. That's the one I just talked about. But there's also neutralization of the exotoxin. This too is neutralization, but by a different means. Watch this. The antibody will bind to the exotoxin the bacteria secretes to do what? Watch. Precipitate it out of solution. Wow. This guy is circulating in your blood, the exotoxin. The antibody binds to it and precipitates it. Then that means it can't go anywhere. So this is neutralization of the exotoxin by precipitation. You gotta catch these two. They're both neutralization. Neutralization of the virus by attaching to by the antibody attaching to the spike protein, preventing the virus from ringing the doorbell. Adsorption. But you also can neutralize the exotoxin. Those exotoxins that would otherwise stimulate the T cell directly, that would bad super antigens, right? You would produce an antibody against this exotoxin to neutralize it by precipitation. Now, if you remember, some of these exotoxins are so bad, you may not have a chance to develop this antibody. So what do? Do you remember? What do we do with those exotoxins to protect you? Wait. You gotta speak up. Say it. Excellent. What is that?
Is like when they create the vaccines, right?
Okay, they create a vaccine. What's the nature of the vaccine in this case?
It has like the piece
Of the
the exotoxin.
exotoxin. Is it a piece or a denatured exotoxin?
Denatured.
There we go.
Yeah.
So we would take the exotoxin, we would grow the bacteria in large quantities, on purpose let it secrete the exotoxin in a lab, not inside you. Get the exotoxin, denature it somehow, typically by heating. That denatured exotoxin is called a toxoid. Because we would inject you with that denatured exotoxin to get your immune system to produce these antibodies before you contract the real thing. Otherwise, some of these exotoxins, they're so bad, you're not even gonna have a chance to develop this. Because you develop this upon primary exposure. If you survive, then you'll have the hopefully lifelong protection against it. But again, some of these exotoxins are so bad that you can't run that risk. So you get a toxoid injection, get your body to produce the antibodies against it, and now your antibodies are ready in case they see the real thing. What are they gonna do? They're gonna bind to it, precipitate it. What did we call that? Neutralization. So, did the antibody kill anything? No. The closest it came to killing is that first one by indirectly interfering with everyday business, right? Okay, this one here. When we talked about vaccines, this is what we're talking about. Watch this. Every time you get sick, the first time you get sick, there's gonna be a latent period. This is the time when your B cell and your T cell communicate with the antigen presenting cells. They're getting activated, they're getting clonally selected, clonal expansion, they're trying to figure out what's going on. And that's the latent period. And then initially, IgM is produced. Upon first exposure, you are initially protected by IgM. Then a little later, IgG is produced. So that's typically what would happen to all of us when we first come in contact with a pathogen. And I hope we survive. What do we have now at the end of this? In this period of time here, what do we have? We have memory cells. Because during this period, you stimulated the T cell, they proliferated, they differentiated into the regulatory T cell, all the different kinds of helper cells, and the memory T cells. You activated the B cells, they proliferated, they went and produced regulatory B cells, the plasma B cells that release the antibodies, and memory B cells. So at the end of the first exposure, also referred to as primary exposure, what are you left with? The memory B cells. I hope for life. Now watch. The next time you're exposed to the same pathogen, there will be no latent period. Or maybe very little tiny latent period. But no latent period, because your memory cells will be like, oh, we've seen this before. You don't have to clonally select. You don't have to differentiate. You don't have to do this. We already did that before in the primary. Your memory T cells and B cells will immediately activate. And look what you get. A huge response. Look at it. It's mainly IgG as well. Remember me saying this? Upon secondary exposure, it's going to be mostly IgG protection. But look at the curve. Huge response. In the secondary response, you have two things going on. Number one, immediate fast response. Number two, robust response, means huge response. Like anything else in biology, that too has a name. And it's a weird name. Anamnestic. That's an S, hang on. Anamnestic response. Also referred to as the secondary response. But you gotta understand what is the characteristic of a secondary response. Fast and robust. Robust means huge. Fast and robust, just like you see here. There's no latent period, and it's a huge response, mainly protected by IgG. And that's a normal infection. Normal primary infection, secondary infection. Now, I told you there are some infections that we can't risk this for. I mean, you won't be able to survive long enough to produce memory cells. Those are the ones we vaccinate for. We give a part of the antigen to mimic the primary response. We trick the immune system to thinking that you're sick when you're really not sick. And what are we after? What are we after with the vaccines? What are we after? Huh? What are we after when we vaccinate, based on what you understood here? To activate, yeah, but what are we after? What are we trying to get? What are we trying to achieve when we vaccinate, based on what I just said? Okay.
Killing the pathogen?
I said this is a normal infection. Normal infection that makes you a little sick, but at least you survive. Primary infection, you get the latent period. That's the time when your body is trying to figure out what's going on, presenting the cells, clonal selection, clonal expansion, the differentiation, and everything, right? What's happening, that's trying to fight the first infection. All the things that are happening here, you're trying to fight the current infection. But how are you protected from that point on? Who's protecting you?
Yeah,
Huh? Who's saying that? Memory cells. That's what you're after when you vaccinate. When you vaccinate, you're tricking the immune system to think that it's sick when it's not, when you're not sick. To produce this effect, but what you're really after is the memory cell. The memory cells, the B and the T cell memory cells will remember the antigen that you injected as the vaccine. The next time the real thing comes in, you're already prepared. You already have the memory cells against it. And you're gonna get an anamnestic response, which is what? What are the two characteristics? Fast and robust. Because otherwise, you may not survive that pathogen. That's the whole idea. Okay, last couple of slides. There are different kinds of vaccines. There are different ways of vaccinating. You can get what's called a live attenuated vaccine, meaning, and you'll probably have to sign a piece of paper to agree to getting a live attenuated vaccine injected in you. But what does that mean? Attenuated. Yes, it is alive. But attenuated means weakened. They took the bad parts out of it and they're using it more to achieve that molecular weight criteria. Remember, high molecular weight? I mean, there's nothing better than using the whole virus. You see? You want to achieve high molecular weight, use the whole pathogen. That will guarantee you, number one, multiple antigens, various sizes, a lot of epitopes to choose from. But you can't inject the real thing in there. You have to attenuate it. That means weaken it somehow. There are ways of doing it. There are advantages and disadvantages to everything. The advantage of using a live attenuated vaccine is the fact that it's alive, that it multiplies. And what will that do? It will keep your immune system activated. It will keep it stimulated because it's alive. So you get a much better immune response with a live attenuated vaccine because it's alive. Your immune system has to keep working, trying to kill it. So you get a better response. There's a disadvantage to it. What is that? It's alive. Wait, hang on. Well, you gotta understand something. Because it's alive, and that's why you're signing that piece of paper, it can mutate. Now, the chance of it happening is very little. But it can mutate, technically, because it's alive, and become really bad for you. And when you sign that piece of paper, after you read the 15 pages of very small 4 font, nobody reads it. You need to realize that. Yes, I know the benefits, and yes, I know the disadvantages. But overall, they work really well, actually. If you don't want to mess with that, then you obviously go to this side, inactivated. We already talked about the toxoid. That is a denatured exotoxin that we use as a vaccine. We can take pieces of protein. We can attach it to things, conjugate it to make it bigger. We can take the antigen, remember with the carrier, attach it to something to make it, to simulate the molecular weight criteria. All of those things can be used as a vehicle for vaccination. Okay, route. This one is easy. You can take it through your mouth. What would that be? Oral route. You can take it through your nose. What would that be? Intranasal. Under the skin. That means a shot. Subcutaneous, in your muscle. Do you remember what COVID was? Which one was COVID? No, it was not subcutaneous. Didn't your muscles hurt for a little while? It was this one. COVID was more intramuscular. Now they have like the flu vaccine. They can deliver it through intranasal, but it's not as good. You have to deliver higher dose of it to get it to have it more active. You don't need to know this. We know every vaccine has side effects. You can't avoid them. And this is just a reference for your future studies in nursing school. How's that? But I will not test you. The last slide, finally, right, is something called herd immunity. You're gonna feel like a cow today, right? What is herd immunity? Herd immunity is what they were trying to achieve with COVID-19. Herd immunity is all about getting everybody vaccinated. Everybody meaning the herd. See? I don't like this term. I don't know why they use this word. But we are typically the herd. That means everybody. When everybody is immunized against something, yeah, then the next person born into that population without vaccination is automatically protected. I mean, that's pretty cool. If everyone in this room is vaccinated against a pathogen, that means we're all protected against, that means we all don't have it. The next person that walks into this room without being vaccinated is automatically protected. Why? Because who is he gonna get the pathogen from? He gets it from another person, right? Well, but we're all good. And that's what they were